The ribosome. Now our ribosome has a large subunit and a small subunit. And our messenger RNA is going to go right here. So the ribosome attaches to the messenger RNA. So the ribosome attaches to the messenger RNA. What is the ribosome made primarily of?
RNA.
Ribosomal RNA. So we know that this is made primarily of RNA.
What's being released?
That's the messenger RNA going up. So we end up down here with exactly the same messenger RNA we started with up there. So I'm really just rewriting it. So all I did is rewrote our messenger RNA here. So our messenger RNA left the nucleus, ribosome attached to it. Here's our message. Well, now what needs to happen? Yeah, we transfer RNA needs to bring our amino acids, right? So out in the cytoplasm, we've got some transfer RNAs. And those transfer RNAs have an amino acid at one end and an anticodon at the other. This is a transfer RNA, just in case. And there are bunches of those around. And no, they don't actually look like that, but it's good enough for our purposes. So the transfer RNA's job is going to be to bring the amino acid down to the ribosome. So does that mean I'm going to start here and start putting in my amino acids? Do I start at the beginning and go left to right? No, what do I need to find? You need the initiator codon, right? Do we have an initiator in here? Sure we do. We get to AUG, that means start. So we're going to ignore what comes before it. What comes before it is irrelevant. But we're going to start there. And then if we're starting there, now the one thing I can't show you in this diagram, guys, is when this is happening, the ribosome is actually sitting right over where that initiator is. The ribosome is actually what holds the transfer RNA and the amino acid in place to allow the bond to occur. But I can't make this move because it's not animated. But if we have our codon coming in, we've got our transfer RNA. Transfer RNA's got an amino acid on top. What did we say AUG codes for? It is start, but it also codes for methionine, right? Here's our methionine. Now, how did we say that this transfer RNA is going to know to put methionine there? How does it know that's where methionine goes?
So 1-to-1 correspondence between the protein and the.
No, not quite. What's on the other end that we still need to deal with? The anticodon. And what did we say about anticodons and codons? They are what? Complementary. So, what's going to be the anticodon that's complementary to AUG? What goes with A? A U. A, C. So that's the anticodon. That's how methionine knows to wind up here. These transfer RNAs are just going to kind of be moving around the cell. But as they get here, now there's a temporary attraction. They're complementary. They're going to want to base pair. The ribosome will be there to hold it together long enough for the bond to occur, but the initial stoppage of that transfer RNA that's moving around is the complementary nature of the codon. So once we do this, then we go over to the next one. Our next codon is CCC, so again we're going to have another transfer RNA come in. What's complementary to CCC? All right. Now here's where you want to avoid the mistake. It's the codon that codes for the amino acid, not the anticodon. So CCC codes for what? Say again? Proline. So proline's going to come in here. We're going to make a bond between those two amino acids. Once that bond occurs, this transfer RNA is going to go away. It'll wind up looking like this. That goes away, so now it's a transfer RNA with no amino acid. It will go pick up another one and come back. So that means we would actually have this transfer RNA with proline attached still. Proline would be attached to methionine. Now we go to the next one. Another transfer RNA. Another complementary anticodon. And what does UUU code for? That's the anticodon, but what amino acid does it code for? Phenylalanine. Oops, right in the wrong color name. So phenylalanine, again another bond. Now we go over to GAU. GAU has as its complement CUA. What does GAU code for? Aspartate. Also known as aspartic acid, but it's okay. ASP. Again, we get a bond. Well, then we get to UAG. What does UAG code for? Stop. This goes back to the question he had asked earlier. UAG means stop. So what's going to happen is once we get to the stop codon, it's going to release this protein. And now we're going to have a protein that's primary structure is methionine, bonded to proline, bonded to phenylalanine, bonded to aspartate, and that's going to be the primary structure of our protein. That protein will then go on and get modified by the Raphiar and the Golgi, develop its secondary and tertiary and quaternary characteristics. But that makes our whole protein. So again, you can see that the DNA coded for the message then determines how the protein is put together. So DNA, which is your genetic material, it controls who and what you are by controlling the proteins that you produce. All right, questions about that? Anything about this that doesn't, yes?
What did you say about the UAC to the far left? The one that's floating around?
Oh, it's just, once this is bonded, it lets go, this transfer RNA will go out, get another methionine, and eventually come back, and then go out. So each of these come and go, so you wind up first with this one attached to this, attached to this. Then these two are attached to this one, held in place by this. Then these four are attached here. When it gets to stop, it releases this. The transfer RNAs will go out and get new cargo to bring back. So lots of ways to transfer RNA. It's really like a trucking company. Goes out, gets material, brings it to the site. Goes out, gets material, brings it to the site. Yes?
How did you get UAC again?
Because what's complementary to A?
Oh, okay.
Is U. A is complementary to U, C is complementary to G.
Oh, but I'm saying that's how it makes sense.
This gets that way because this determines the anticodon. The anticodon is
Talk about
complementary.
the little guy. The little-
This was him.
From the, oh, okay.
This and this are exactly the same thing. Yes?
So these amino acids are pumped into the cell?
The amino acids are in the cells from what you eat.
Okay.
You eat protein, your body breaks those proteins down into their amino acids, they circulate in your blood supply, your cells then take them and rearrange them into your proteins. Because if you go home tonight and have a hamburger, cow protein doesn't do you any good. You can't do anything with cow protein. You take it apart to amino acids, now you can build it up into people proteins, and then it can do whatever you want. So that gets back to what we were talking about when we were talking about metabolism and anabolism. You consume food, you break it into small pieces, you then rearrange it. Yes?
What's the amino acid and what's the CUA acid?
This is PHE, phenylalanine, and ASP, which is aspartame. Yes? Yep, go ahead.
What did you write on the top? Like the orange thing, you wrote something on?
Oh, it just says nucleus. That was just telling us the orange thing was the nucleus. Yeah, RNA is the main ingredient in ribosomes. Yep.
So the protein just simply breaks off, or those,
Yes.
so it just breaks off once it hits stop, basically?
Yes. Breaks off, and now we have a primary protein in the ribosome, which then will go to, could go to the rough ER, could go to the Golgi, to be further modified before it's ready to actually be an active protein.
And those tRNA just go back to,
Yep, they just go back and forth, scoop it up, transfer RNAs, bring them to the ribosome. All right, questions, comments, issues?